So we're now going to take a look at the mechanisms by which the body assesses when the sodium chloride levels in the blood are too low, and how it activates a series of pathways that will help you conserve the existing sodium chloride that is already in your body. Now, this is basically putting together a bunch of pieces that we've already seen into one story. So some of this will be new, but a lot of it will be rehashed from previous lectures. So our goal is to conserve as much sodium and chloride as possible if our sodium chloride levels are too low. And there are really two branches to this story, one of which is going to occur with the filtrate itself, and the other of which involves blood pressure. And there is some crosstalk between these two branches. So if there's too little sodium chloride in the body, and we know that sodium chloride is concentrated in the extracellular fluid, then there's also going to be too little sodium chloride in the blood. And since filtrate, at least initially, is simply a reflection of what is in the blood, if I have too little sodium chloride in my blood, then my filtrate is also going to have too little sodium and chloride. So let's go ahead and add that. Now that filtrate is going to move through the proximal tubule, where reabsorption of sodium and chloride happens on autopilot. The amount of sodium chloride that is removed is a percentage of what is in filtrate. And the same is true for the ascending loop of Henle. So what that means is that if my filtrate already starts out with too little sodium chloride, then by the time my filtrate moves through the proximal tubule and the descending and ascending loop of Henle, then I'm also going to have a paucity of sodium and chloride in my filtrate when it reaches the distal tubule. So by the time the filtrate reaches the distal tubule, it's also going to have a low amount of sodium and chloride. Now this is important because sodium and chloride are actually directly measured in special sensor cells that exist in the early part of the distal tubule, where the distal tubule crosses the afferent and the efferent arteriole in what is known as the juxtaglomerular apparatus. So what happens is cells in the juxtaglomerular apparatus sense that there is too little sodium and chloride. And these cells, they're the macula densa cells, but you don't need to know that, are going to talk to the granular cells, which you absolutely need to know, and tell those granular cells to secrete renin. So renin is going to be secreted from these granular cells into the bloodstream and activate the renin-angiotensin-aldosterone pathway. And we've covered this in a prior lecture, so we'll quickly review it here. Renin is an enzyme that is going to take angiotensinogen, which is a plasma protein, and convert it into angiotensin 1. And then another enzyme called ACE is going to convert angiotensin 1 into angiotensin 2, which is actually a small peptide. And we know that angiotensin 2 has three major effects, two of which I really highlighted in a previous lecture. And one of those effects is to stimulate secretion of aldosterone. And it is the adrenal gland, specifically the outside of the adrenal gland, the adrenal cortex, that secretes aldosterone. Now, we know the end of the story at this point. We know what aldosterone does. So we know that aldosterone is a hormone that is going to target distal tubule cells and cause those cells to insert more sodium-potassium pumps into the basolateral membrane. And we know that is going to increase the amount of sodium that is reabsorbed. In fact, we can reabsorb up to 100% of our sodium. Will also increase the amount of potassium secretion, though that's not really relevant here. By increasing the amount of sodium that is reabsorbed, you put this back into your bloodstream. So we are conserving our sodium in chloride, and that means there is less that is being excreted. So feel free to pause and rewind as needed to make sure that you get all of this down. I'm going to go ahead and zoom out a little bit so you can kind of see how big this is. Okay, so we went through a whole bunch of stuff in the filtrate, and now we're going to go ahead and talk about blood pressure. And there's going to be some crossover here between the blood pressure and the filtrate columns. So if there is too little sodium and chloride in the body, then we also expect that there's going to be too little sodium and chloride in the blood, and this will reduce our blood volume and therefore reduce our blood pressure. So blood pressure is actually felt in a number of different places. I mentioned two major baroreceptors in the aortic arch and the carotid sinuses. And so these are going to sense lack of stretch. And those baroreceptors are sensory neurons that are going to feed into the autonomic nervous system. And that's basically going to trigger the sympathetic nervous system, which is synapsed into the afferent arterial cells and therefore also the granular cells. It's going to go ahead and trigger secretion of renin. In addition, the afferent arteriole itself, which we referred to in a previous lecture as being able to kind of self-regulate with a myogenic stretch reflex, also has ways to sense stretch. And therefore, if it doesn't sense enough stretch, it will also secrete renin. So there's actually 3 different triggers for renin secretion. And this is not going to be on the test in this detail, but you should know that one trigger for renin secretion is going to be from the juxtaglomerular feedback from the tubular cells, and then 2 different types of lack of stretch, one being from the baroreceptors in the carotid artery and the aortic arch, and then the others just being in the afferent arteriole and the kidney itself. But effectively, lack of stretch is going to be a trigger to secrete renin. Okay, and hopefully you can also see that angiotensin II, which is a powerful vasoconstrictor, will assist in helping to increase our blood pressure by tightening up the blood vessels. Now, this right here is pretty much our typical flow chart when our sodium chloride levels are too low. But I want to talk about an extreme example. And I'm going to put this part in red because it really only happens when our blood pressure gets dangerously low. The sympathetic nervous system can do additional things beyond triggering renin secretion. And so I personally refer to this as kind of a sympathetic override. And I have mentioned it in a prior lecture when talking about ways that we can control GFR, or glomerular filtration rates. So this sympathetic override, again, is only going to happen when blood pressure is dangerously low, not just a little bit low. When the blood pressure is dangerously low and there's almost no stretch, then the body recognizes how important it is to keep water and blood in the blood vessels. Under typical times, around 20% of our cardiac output can be in the kidneys. That's a huge amount of our blood. 1/5 of our blood is in the kidneys. And what's it doing in the kidneys? Well, a lot of it is being filtered and then reabsorbed and filtered and reabsorbed. But while it's in the kidneys, it's more or less out of commission. And so if your blood pressure is dangerously low, you cannot afford to let that blood spend time in a place it doesn't need to be. If you have super low blood pressure, you are going to try to restrict the blood flow into the kidney so that the cardiac output is much, much, much less, and also restrict the glomerular filtration rate so that you're not spending time with blood as filtrate that you may not even recapture all of. So in other words, your body is going to sacrifice blood chemistry balance for blood pressure on kind of a short-term emergency measure, and that is the sympathetic override. So in the sympathetic override, what basically happens is we vasoconstrict a variety of renal arteries. This is going to reduce the amount of blood going into the kidney in the first place, and we also vasoconstrict the afferent arteriole. And that is going to reduce the glomerular filtration rate. So both of these actually aim to reduce the amount of blood in the kidney and reduce GFR. If you reduce GFR, then you have less sodium and chloride as filtrate. Remember, when it's in filtrate, it's not being used in a useful way, and therefore you have a way to conserve more sodium and chloride. You should be able to understand this entire pathway, and you should be able to draw the converse of this pathway. So I will briefly walk you through what I mean by the converse of this pathway. Imagine that instead I ask you, what happens when we have too much sodium and chloride? What's going to happen? And I'm just going to briefly point out sections and tell you how you would change them, and you'll see the pattern. If you were to write this, you would say the filtrate has too much sodium chloride, which means that the filtrate ear also has too much sodium chloride, which means that the cells are going to sense too much sodium chloride. So what that means is that the granular cells are told to stop secreting renin. If you don't secrete renin, then this process doesn't happen, which means this process doesn't happen, which means there's no angiotensin II produced, which means there's no aldosterone secreted. When aldosterone is not secreted, the target cells in the distal tubule are going to remove sodium-potassium pumps, which is going to decrease sodium chloride reabsorption, and you are going to have more salt excreted. So hopefully you can kind of see how you can do the reverse. And the same is true on the blood pressure side. We're going to have increased blood volume, which is going to be increased blood pressure, which is going to become baroreceptor sense stretch, which means the sympathetic nervous system does not talk to the granular cells. And obviously there's no converse to this. This doesn't happen. Okay, so we have now covered the mechanisms by which the salt balance is achieved in our body. We are now briefly going to talk about water balance, which luckily is a lot easier. So let's talk about what happens when our body is dehydrated versus when we've, let's just say, ingested 2 liters of water and our water reserves are too high. Let's start with being dehydrated. If I am dehydrated, I expect my blood pressure to be low, but I expect my osmolarity to be around 290. So remember that we have osmoreceptors in the hypothalamus, that are going to detect that and basically tell a neighboring section of the brain called the posterior pituitary gland to secrete antidiuretic hormone. And we know the rest of the story, so I'm not going to spend too much time going through it. Antidiuretic hormone is going to target what? Exactly, the collecting duct cells. And it's going to do what? Perfect. It's going to add more aquaporins, which is going to result in an increased amount of water reabsorption and therefore a decreased amount of water excretion. All right, we can just do the converse. So if I ingest 2 liters of water, I expect my blood pressure to probably go up, but most importantly, my blood osmolarity will go down. And that's also going to be sensed by osmoreceptors. They're going to sense this, and they are going to tell the pituitary gland to do what? They are going to tell it to stop secreting ADH. When you stop ADH secretion, the ADH levels in the blood start to fall, and the collecting duct responds to this by removing aquaporins. And that is going to decrease water reabsorption and increase water excretion. So if you were able to follow this, great job. So that concludes our water and salt balance set of lectures. I encourage you to go back through and try to draw these out from memory. And you really don't need to memorize it if you can understand the story and what makes sense. All right, thanks, and I'll see you guys in the next lectures for endocrinology.